think we have a full schedule, so I think we should probably go ahead and get started and as people start trickling in. So my name is Olivia Viveros, and I am the MESA director here at College of San Mateo. I wanted to welcome everyone and thank you for joining us tonight on our first Science in Action speaker series event of the semester. Before we get started, I just want to remind you that you could write the questions in the Q&A or in the chat for our panelists, and then they will answer them at the end of their presentation. We have two fantastic speakers tonight who will be sharing their STEM career and experiences. One of them is a community college student or former community college students themselves. And I would like you to now introduce Panos, who is our moderator, who is also a Stanford student pursuing a master's degree in epidemiology. So it's all yours, Panos.
Thanks for the introduction, Olivia. My name is Panos. I use he, him pronouns, and I'll be moderating today's panel. I'd like to thank Olivia and the MESA program for all their help in putting together this speaker series. I'd also like to thank Sean and Teni for serving as our first pair of panelists of the year. So today's panel will start with Sean and Teni each spending about 10 to 15 minutes talking about their career paths and current work, and then we'll transition to about 30 minutes of Q&A. So please feel free to submit questions at any time by clicking on the Q&A button at the bottom of your screen, and then I'll read them out loud at the end for Sean and Teni to answer. So now we'll just jump right in with Teni, and I'll hand it off to her.
All right. Hello, everyone. I am going to start sharing my screen. Okay. Is my screen showing?
Yes.
Okay. Great. All right. Thank you so much to Olivia and Panos and all of you who are attending for being here today. My name is Teni Anbarchian. I am currently a medical student at Stanford University. And I'm really excited to be talking to you today, especially since I also went to a community college, and I hope that we can perhaps relate in some of our stories today. All right, so the image that you're seeing here, I'll explain more about it later, but just a teaser, it's a slice through the liver, and all the white kind of parts that you're seeing, these are liver veins. As you can see, there are branches here, and the small colorful dots that we're seeing here are some cells that we labeled with different colors. So I'll talk more about this later, but just wanted to explain what this picture is here. All right, so I wanted to start with a little bit about my background, more like the cultural and geographic background. I was born and raised in Iran, in the Middle East. However, my ancestors are from a country in the Caucasus called Armenia. It's a small country just to the north of Iran. And we unfortunately weren't taught a lot about when and how long ago and how my ancestors immigrated to Iran. But what we think happened is that they were brought to Iran a few hundred years ago, and they settled there. There is a community of Armenians there. And so I was born there in the capital of Iran, Tehran. And I grew up among Armenians and also Iranians. So culturally, I relate to certain parts of both cultures. I grew up, me and my Armenian kind of fellow Armenians, we grew up as minorities in Iran. We're both cultural and religious minorities. So I can understand some of the challenges that people might go through when they feel like they're not represented in the country that they live in. So I'm happy to talk more about this if anyone is interested. But that said, the challenges that we face also teach us some lessons. And I think that the cultures that we share and the bits and pieces of cultures that we carry with us kind of make our experiences more unique and helps everyone bring something new to the table. So I grew up in Iran, as I mentioned, and when I was 17, we were very fortunate that we were with my family. We were able to migrate to the U.S. and specifically California. In Iran, I was very interested in biology in general, but my grades in biology were really bad. I was much better in math and sciences, and my high school counselors would often tell me that I should give up on my dreams of wanting to become a scientist. And the reason I wanted to become a scientist was because I just was really amazed by how cells and the body in general work. But like I said, I was really bad at biology. And besides that, I guess besides that, in Iran, there were not a lot of opportunities to become a scientist. I didn't know any scientists, really. On the other hand, my parents were both educated, and I was really privileged to have that sort of support at home, where I was never told that I cannot become a scientist, even though the opportunities were not there. My parents always respected the time I spent on studying, and I think that was a really big privilege for me growing up. So when we immigrated to the US, I really wanted to become a scientist, and I thought that I have my path set. But when we came here, I realized I don't know anyone to help me navigate the system. I didn't know where to go, so I went to a local high school, and I wanted to sign up, but they said I was too old to start high school. To this day, I still want to go to high school one day, attend American high schools, because from what I hear, they're really fun. You might disagree, but one day I'll try it out. It sounds weird, but we'll see. Anyway, I'm kidding about that. So after I realized that, okay, I cannot go to high school, which means I cannot graduate from high school here. I won't have that degree. What do? I don't know how to go to college. So I registered at a community college. This was in Los Angeles, California. Glendale Community College was where I went, and I signed up there. Mostly classes that I took in the beginning were English classes. My education growing up was in Armenian and Farsi, so I didn't know. I knew some English, but not enough to study math and biology and social sciences. So those are a lot of the classes that I took in the beginning, and it was a difficult transition. I still didn't know how to pursue my goal of becoming a scientist, and I didn't know anyone else in my community college who wanted to do that. I had a lot of friends that I had met in college, and they wanted to pursue some professional careers, like they wanted to become a dentist or a doctor. And the path was more clear for them. So I was like, okay, we all have to transfer at some point. So what are some resources that we have for transfers? And those resources were also kind of scarce. So I joined the Alpha Gamma Sigma Honor Society, and I became the vice president of academic events. And with help from the group, we started some transfer information sessions where we gathered information on transfer applications and places to transfer. And we held those sessions, which helped both myself and all of my peers. We also had faculty speaker series that we organized, and those also helped us really get exposed to some scientific, to the scientific world a little bit, but still we didn't have access to research opportunities, or we didn't have role models really. I still didn't know anyone who was an active scientist in community college. And just to add here real quick that I also worked through college because as many of you may know, a lot of community college students are part-time or full-time workers. And that was an important thing for me as well as a new immigrant. I needed to both support myself and my family financially. So that was another challenge. So finally, after about 2.5 years at community college, I transferred to UCLA. One of the main reasons why I went to UCLA was because it was close to family. Having recently immigrated, I didn't want to leave them and be far from them. So this was a really great option for me. I chose to study molecular cell and developmental biology. And since I had no experience in research, and I noticed that a lot of my fellow UCLA classmates had already been in labs for a couple of years, I minored in biomedical research, which was a really amazing program. I want to give a shout out to the biomed research minor because it really prepared me for grad school. And how they did that was they quickly helped you get placed in a lab. They helped us understand scientific literature and be able to critically analyze it. We also practiced writing and speaking about scientific data. They created a great community for us, and they presented us with opportunities for earning scholarships. So at UCLA, I joined Dr. Karen Lyons' lab. She was a great mentor for me, and you'll probably notice that a lot of times where speakers present their work, they'll show pictures of their mentors, and that's just really because mentors make a very big impact on our lives. If it wasn't for Karen, I wouldn't have applied to Stanford for my PhD work. Karen and also a graduate student mentor that I had, Diana Rigger, both of them were really important in my journey. So let's see. So I transferred. So I finished working my studies at UCLA. I stayed there, worked a year there, and then I applied to the PhD programs, and I was accepted into Stanford University, the developmental biology program. And I was there for about 7 years. I recently graduated. I was and still am in Dr. Roel Nossa's lab. We work on a lot of developmental biology questions, and if you're interested in what that means, that's basically just it's a very broad topic, but how do cells divide and grow and create organisms that continue growing until they become a functional organism, like an adult human being. And for the interest of time, I won't go into the details of this, but if you're interested, you can visit Stanford's developmental biology website, and you can see how developmental biology includes a lot of diverse topics, from stem cells to cancer to regenerative medicine. So I'll very quickly introduce what I worked on as a PhD student in Stanford. I specifically studied the liver as an organ, and what I was interested in was how does the liver form and grow? So to understand this question, we worked with mice. And the very basic answer to this question is that cells have to divide to make more cells in order to grow the organ. And that's kind of a cell cycle, where a cell goes through the cycle and becomes two, divides into two. So that's a very basic question in biology that we ask. It's very relevant to cancer and regenerative medicine. So one way that we have to look at cell division is using microscopy, and through some kind of complicated genetic assays, we can label cells with different colors in a tissue that you like, and then follow them over time. And I don't want to go into the details of this, just to show you that scientists can use cool microscopy and genetic tools to make things that we cannot see become visible to us, like cells and tissues. And panels, if I'm really running late, please let me know, because I think I've been speaking for a while now.
Yeah, you're all right.
Okay, thank you. All right, so different ways that we have for looking at how tissues work and cells divide is that we can identify a gene that we think is really important. In this case, we looked at the gene called TBX3. We think TBX3 is really important in development of the liver. And one way that we use to answer that question is by deleting the TBX3 gene from the liver, and then asking, okay, what happens now if you don't have this gene? What happens to the tissue? So the white spots that you're seeing here are nucleus of the cell where the DNA can be found. And that's where we're also seeing some TBX3 protein. This is a normal mouse liver section. So this is like a very small window into the liver. And then when we delete TBX3 on this image, we can see that those white spots are gone, which means that my experiment worked. I have eliminated TBX3 from the liver, and then I can ask, okay, what happens to the cells now? So we can also label our cells so that we can see them better. When we have a normal liver, for example, we can label all of our nuclei with some green fluorescent protein. When we get rid of TBX3, we can see that the nuclei of the cells have become much larger, which means that these cells have accumulated DNA in their nucleus, and they become very big cells. When cells accumulate DNA, they can no longer divide. And that means that they can no longer contribute to the growth of the tissue. So through my PhD thesis, what we found was what are some regulators of TBX3? What are some signals that make cells make TBX3? And how does TBX3 control division of the cells? I won't go into the details of this, but it matters because by studying this, we can learn about how tissues grow, what makes the liver do its job right, or what happens when things go wrong. It helps us understand liver regeneration better. The liver actually has a really good capacity for regeneration. And it gives us insight into carcinomas, cancers of the liver. It also helps us understand how we can grow more liver cells in culture. Okay, so finally, I wanted to tell you that when I finished my PhD program, or before finishing it, I realized that I actually really wanted to pursue medicine. And so I applied to Stanford and several other programs for MD, and I recently started medical school a couple of months ago, and I misspelled medical school here. But the goal is to do a few more years of studying and then residency, and finally, hopefully become a physician scientist where I can both see patients and do research in the lab. And this is just a really happy picture of me a couple of months ago during our white coat ceremony. Okay, in the interest of time, I would like to end here. I will not go through my other slides. I'll just let you know that science is fun, and there are a lot of ways to talk to people about science and promote science. And we are actually now we actually now, in collaboration with your college, we have a research program at Stanford that you can apply to. And this will place you in labs that you can do research in. And finally, thank you so much. You can reach me via email or on Twitter if you'd like. I'm not very active, but I follow a lot of physicians and scientists, and I can introduce you to people if you're interested. And yeah, I'll end here. Thank you so much.
Thank you so much, Teni. That was a wonderful presentation. We already got a message from one of the attendees. I love your story and passion for science. Thank you for sharing this information.
Thank you.
We'll still do Q&A at the end. Feel free to submit your questions in Q&A. We already have one. But now we're going to move to Sean's presentation. Sean, take it away.
All righty, great. Thanks, Panos. Okay, great. So hopefully everyone can see my screen here.
Yes.
Great. So my name is Sean Yamada-Hunter. I'm currently a postdoc in the Mako lab at Stanford, where we study cell therapy and designing new cellular therapies for cancer treatment. And it's an honor to be selected as a speaker for the Science in Action series. So thank you to Panos and Olivia and all of the other organizers. So I won't dive too deeply into my science that I do, but I'm happy to answer questions about that during the panel section. But I'll kind of tell you about my path and how I ended up as a postdoc at Stanford. There's actually a number of overlaps with Teni, especially down the road in terms of our undergrad and grad school. So I was born in San Diego. I've actually lived in California my whole life. I've been in SoCal as well as NorCal, and certainly love both places, though San Diego has a special place in my heart. It's kind of funny to hear Teni's story about not really knowing any scientists growing up, or not knowing scientists until kind of later in her career, and yet always wanting to be a scientist. And certainly for me, I've also always wanted to be a scientist, although I have known a scientist for a very long time. And so growing up, kind of a second home was this building, which some of you might recognize. It's famous for its architecture, but this is the Salk Institute for Biological Studies. It's a research institute in San Diego, located actually right next to UCSD's campus. And I spent a lot of time here growing up because my dad, Dr. Tony Hunter, was a professor there. And so like a lot of kids growing up, I wanted to do what my parents did. My parents were both scientists, so I always wanted to be a scientist from a very young age, even though I didn't really know what it entailed. Certainly was a role model to kind of see science done at a high level. And so my dad certainly was a role model. He's well established in his field, studying cell biology and cancer biology specifically. His kind of claim to fame, and a word that I knew since I was like 5 years old, is the discovery of phosphotyrosine. So tyrosine is one of the 20 amino acids, which are the building blocks of cells, or of proteins. He discovered that tyrosine is one of 3 amino acids that can have this phosphate group added to it. This is a way that cells communicate, that they send signals. And it turns out to also be a way, this tyrosine phosphorylation, that cancer cells send signals, and that cancer cells kind of grow and divide. And so it ended up being a big discovery for the development of cancer therapeutics, and certainly work done by many others led to the discovery of this drug called Gleevec, which was one of the first really powerful targeted cancer therapies that ended up being very potent for treatment of leukemia. And so this was kind of the environment that I grew up in, kind of seeing a lot of science, meeting a lot of scientists, and certainly being inspired by my dad's work to go on and be a scientist, although he was very frank with me that science is not always as rosy as it might seem from the outside looking in. I was able to work in my dad's lab for a few summers and kind of get a flavor of research, but it wasn't until I went to undergraduate that I really dove into being a researcher. And so like Teni, I also went to UCLA. We're both proud Bruins. In fact, we were there at the same time. I was there from 2009 to 2013, although I don't think we ever met each other. I majored in biochemistry, so kind of knew that I wanted to be a scientist and just applied for biochemistry. It seemed kind of to measure up to a lot of my interests, although I don't think I really knew what biochemistry was necessarily going in at the time. While I was there, I applied to a number of labs and was lucky enough to be accepted into 2 over the course of my career there. But the first was working with Hanna Mikkola, whose lab studies hematopoietic stem cell development. So this is the development of the blood system. And so I was able to work there for my sophomore year, kind of helping them as they really pushed out a big story for the lab. And so it was kind of a heady time to be there and to be doing experiments that were really important for this work and kind of seeing how really fast-paced science is done. But after the year, I decided that kind of the developmental biology wasn't so much what I wanted to do, and it wasn't really in line with the biochemistry I was learning. So I actually took kind of the year off as my junior year, but I came back to doing more research as a senior and ended up applying and working in the lab of Heather Christoph. This was kind of my introduction into cancer research and studying how cancer cells grow. The Christoph lab studied cancer metabolism, so kind of how cancer cells utilize and grow from factors in their environment. And so it was a very kind of small lab at the time, but I got to work really closely with a great graduate student, Abby Krall, who kind of really let me have a lot of my own ropes and designing my own kind of experiments. And it was very much in line with the biochemistry I've been doing. And so I think that experience really drove me to say, yes, I do, this vision I've had since I was a little kid, but didn't really know what it was to be a researcher. This was kind of the first time where I was really enjoying the research in terms of kind of having my own projects and moving them forward. So this was my senior year. At that point, I took a year off in between to apply to grad schools. I worked as a tutor. Teaching is still something that I'm really passionate about. So I was teaching a variety of different STEM subjects. And applied to graduate school and was lucky enough to come to Stanford for my PhD. And so I've been here for quite a while now. Like Teni, I came in the class of 2014 in the cancer biology program. And so this is one of the programs in Stanford's Biosciences Hub. The program is really just generally focused on cancer biology, anywhere from basic cancer biology all the way through into translational biology, treating cancer, which is more the area that I've done my research in. And Stanford has just been an exceptional place, and it's been a lot of fun to be up here in the Bay Area and kind of be here for so many years. So during my PhD, I worked in the lab of Jennifer Cochran, who is the current chair of bioengineering at Stanford. The Cochran lab studies and works on a field that I actually had no idea existed, certainly as an undergrad, and even when I came to grad school, called protein engineering. Protein engineering is this awesome field that is based around the idea of designing and engineering proteins to do certain jobs better or more effectively than they might already do. And so the main kind of focus of the lab was to use a process called directed evolution, which won the Nobel Prize actually a few years ago, which is the idea is taking a process of evolution, right, which is small incremental changes that build up over a long period of time, but doing it very quickly over the course of a few weeks. And so in this way, we're able to evolve proteins to have kind of new and improved functions. And the big focus of the Cochran lab, as Jennifer shared during a talk that I went to my first year, where I was totally blown away and decided this was a lab I wanted to join, was in developing cancer drugs. And so that was the focus of my PhD, was developing this new protein drug to treat cancer. Specifically, I was targeting a small kind of cytokine growth factor called the leukemia inhibitory factor, or LIF. I went through the whole process of choosing the target, engineering this protein to bind to LIF very tightly and block it from interacting with cancer cells, and then making the protein and testing it as a therapy in models of pancreatic cancer, being the cancer we're interested in. And so it was a really deep dive into how to make a protein drug, how to treat cancer, and ultimately what's exciting to see an effect, to see a drug that could one day, far down the road, go into patients. And so kind of another fun thing about this that was kind of unique, I would say, is that we were able to publish this study. But if you look at the authors of this study, I was actually able to work with my dad's lab and work with my dad during my time in graduate school. And so it turned out that the molecule we were interested in targeting was something that a postdoc in his lab had been working on. And so we were able to collaborate with him. And that was a very unique experience. I don't think many people get to publish papers with their parents, but certainly something that maybe has kind of come full circle as I was dreaming of being a researcher as a young boy, and then kind of came full circle to actually write a paper with my dad as a PhD student. And so as I wrapped up the PhD, I knew that I really wanted to stay in the area of cancer therapy, but that at the same time as I'd been developing these different protein therapies, there was kind of a novel type of therapy that was emerging that I think has a ton of promise. And so this is cell therapy. The idea is it's a type of immunotherapy, meaning that it takes advantage of the patient's immune system to fight cancer. And in this way, we're able to take cells from the patient and engineer them and change them so that they're able to locate, detect, and ultimately kill the cancer. And so I joined the lab of Dr. Crystal Makel, who came to Stanford during my PhD. And Crystal's an expert in the field of CAR T, therapy, which is the type of therapy that we work on. And I won't go into any kind of details about what I do now, although it's kind of multifaceted, but generally my goal is to apply the kind of techniques and knowledge that I learned from the protein engineering space to engineer new proteins and new synthetic signaling circuits to overcome the challenges facing CAR T therapy, of which there are many. And so whether that's designing new proteins to help CAR T cells recognize and kill cancer, engineering and changing proteins on the CAR T cells themselves, or designing novel signaling circuits, it's all kind of lumped into this idea of using synthetic biology to create new proteins and new reagents that can help these CAR T cells work better and ultimately treat cancer and help patients. And so I'll just give a few takeaways quickly kind of from my scientific journey, things as I was putting this together that I think were kind of like guiding principles for me. And so the first is to be curious. I think it's important to just be open to as many opportunities as possible. I think especially when you're starting out kind of early on and you're thinking about what classes to take and what maybe to major in. I think it's really important to do things that might be a little bit outside of your comfort zone to kind of see if something is interesting and something sparks your interest. Certainly for me, coming to graduate school, protein engineering wasn't even on my radar, but ended up being something that I ended up becoming very passionate about, and that I think has kind of shaped my future career. So I think there's lots of opportunities if you're open to them. And the second is to kind of go with the flow. So I think you don't necessarily have to force yourself down a preconceived path of something, as in like, you have to do X, Y, and Z to get to this step. I think even just listening to me and Teni talk, you can see that there's a lot of different ways to get to ultimately, a career in STEM. And I think kind of allowing yourself to kind of go with the opportunities that are in front of you can open up unexpected doors. And I think in thinking about kind of what you want to do, I think that something that's certainly happened for me is that passion builds with time. I think a lot of people, and I've been going through this myself, try to say, oh, do I love something right away? And I think that oftentimes the more time you spend at something, the more time you spend learning about something, the more you grow to really enjoy it. And I think that this is something where if you give something enough time and you put enough effort into learning about it, a lot of things can be interesting. And certainly science is like that. Almost any question I think can be really, really fascinating once you kind of dive below the surface. And I think something that really was important for me is making sure to take the time to take a step back, whether that was, during undergraduate and after undergraduate, when I took kind of a year off from research to kind of reevaluate and see if it was something that I still wanted to do, or even kind of while you're in the thick of it, making sure to make time for friends, family, and hobbies. And so just to share a few with you, to show that it's not all science for me, a couple of things that I'm really passionate about include sports and the Chargers, big one on Monday night, still a loyal fan from San Diego, going out into nature and experiencing the outdoors, camping and hiking. And kind of my nerdiest passion is crosswords. So I like to write and solve crosswords, and actually had one published in the New York Times recently. So you can check it out if you subscribe. So that's it. Thank you all. And looking forward to hearing your questions and chatting a little bit more.
Thanks so much, Sean. That was a great presentation. We already have several questions for both Sean and Teni. So Teni, feel free to come back on camera and we're going to kick off the Q&A. I'll just ask them in the order that they're asked, but feel free to submit these as we go along. So first one is question for either or both panelists. This goes off one of your last points, Sean. Did you ever get burnout from school, and how were you able to deal with it?
Yeah, so I guess I think, yeah, to my last point, I think was really critical. I think that especially during undergrad, I really tried to manage kind of the load and how much I was working in lab and not trying to take too many classes. Of course, that's dependent on the major and kind of the expectations of the lab and isn't always possible. But I think, making sure you take the time, I certainly tried to do this during grad school as well, to kind of have a support network, to have a group of friends that I could go to and make sure that I wasn't, just becoming too focused on the schoolwork or the lab work. I think was really critical. And I'm sure is very essential to kind of overall, like the way that I still continue to be excited about science. Certainly you can get burned out if you go too much into it. Yeah.
Yeah, I definitely agree with Sean. I think in addition to what Sean mentioned, we, of course, went through undergraduate time when it's a lot of studying, but when you attend grad school, most of the classes are in your first year and a half of grad school. And then the rest, you're mostly really either reading a lot of scientific papers or taking classes that you're specifically interested in. And these are not the same type of classes that you take during undergrad. It's often more interactive or project-based. So you're not always just memorizing things anymore. In addition, I think supplementing your studies with stuff that you like, and also if you are interested in perhaps being employed, like having a job. For me, I really like science outreach. So I'm constantly involved in, talking about science with the public or mentoring people who are interested in science and being scientists or pre-med students. So those things really help charge me for the times that I need to go back to studying. Also try to take breaks. If you have a week of winter break, spring break, take advantage of that. If you're not given that time, you can ask for it. If you're going in between, if you're transferring or if you're going from job to job or things like that, you can always ask to see if you can take a week, a month, a couple of months off to recharge.
Thank you both. Our next question asks, are there research opportunities for all majors? I'm interested in computer science research. And I wonder whether we can use this as a springboard to talk about the interdisciplinary nature of both your research. I know someone else asked you, Sean, could you briefly describe more about how you actually engineer the proteins and mechanisms that were at work and what protein evolution is. So if each of you could just describe a little bit what makes your work interdisciplinary, how do you bring different skill sets from different fields to that work?
Yeah, so right, so Stanford is an incredibly interdisciplinary space, and there's research all over in almost every field. There's not just limited to STEM, but yes, tons of computer science research in the Gates Computer Science building. I had a couple friends during the PhD who were computer science researchers, and there's even computer science research that blends biology as well, using AI to model different structures and things like that. So certainly a lot of opportunities there. In terms of my specific work being interdisciplinary, certainly I think you have anything from kind of the chemistry side, really biochemistry heavy in terms of how proteins actually work, how they interact with one another, into the kind of more cell therapy types like this, kind of more biology, and then also you have kind of the engineering aspect and that mindset. So technically it was a bioengineering lab, and so I think that kind of brings together a lot of things. In terms of how we engineer the proteins, just kind of quickly, essentially you do what nature does, but you speed it up, like I mentioned. And so we make what's called a library of mutant proteins. And so, normally for evolution, mutations would occur kind of sporadically and kind of maybe build up over time. But in this case, we make a bunch of mutations And then we use different techniques to do what's called screening, or trying to identify those mutations that are beneficial. And so we kind of force the protein to change. In fact, the protein that I engineered ended up with 8 mutations that caused it to change and bind with more high affinity. So it's kind of a fun process. You feel like you're making a new protein and evolving it. And you can really, you know, if you set up the right, what's called a screen, you can really kind of develop proteins that do anything as long as you're kind of selecting on putting the right pressures. And so that's kind of generally how that process works. But absolutely, it's very interdisciplinary.
I don't have too much to add to this, except to say, to echo what Sean said, research can be very interdisciplinary in general. Even if you're in biology, you will often need expertise from other fields. For example, I learned how to code, or I improved my coding skills because I needed to analyze single-cell RNA sequencing data, which is a type of experiment that gives you a lot of information that needs to be kind of decoded to get you solid answers. So you have to use basically coding, your coding skills, and you often end up collaborating with people who know more than you do in that field. So there is a lot of space for growth and learning in different fields of science. And specifically about computer science, if you are interested in a lab, in working in a lab, I think the advantage of computer science is that you can probably do research remotely. So if you're interested in like doing this sort of research, look up institutions, universities, and look up their labs. Just read it. So each lab usually has a lab website. You can quickly read about what they do. And if you think you have skills that you can contribute, you know, like if you're good at a certain coding language or something like that, you can send an email to the PI of the lab, that's the principal investigator of the lab, and say, hey, I would like to, you know, gain some experience in research. I really like your idea of like working on this thing. Do you think I could, you know, work with you remotely? I'm really good at this. I might be able to help with the project. Or if you don't have a skill, just say, I would really love to learn how this thing works. And do you think I could, you know, work with you? I think most people are very nice, but they're also very busy. So you might not get responses back right away or at all, but that just means you need to really email a lot of people probably, but give it a try. It might work. It will work.
Thank you both. And Teni, your last answer speaks to a few questions that have come in around the same topic of getting those opportunities and finding good mentors. So what's a way to make myself a good option to be chosen? How do we find good mentors? I often feel alone in my educational journey because of the pandemic, which you also just mentioned, and could use someone to help guide higher education. And do you have any advice for cold emailing with no research experience? So maybe we can talk a little bit more about, first of all, how do you find a good mentor and figure out if someone's going to be a good mentor beyond your interest in the subject matter? And how do you take that first step where you might feel very anxious just coming from classes that don't necessarily teach the research skills?
Yeah, I'll try to answer some of those questions and I'll keep the rest for Sean, maybe. Okay, so I'm going to start by saying that we have recently started a program at our department. It's a community college peer mentorship program. And sorry to be introducing that here, but I think that that's a really good space for you guys, for anyone who's interested, to come and learn more about all of these things, because we can't talk about all of that like in detail right now. But if you attend those meetings, they're happening once a month, and each day will have its own theme, like how to find a mentor, how to email professors that you're interested in working with, how to, you know, prepare a resume or something. So Olivia, I don't have the link with me right now? If you do you think it would be possible?
Yes, I'm looking for it and I will put it in the chat.
Okay, perfect. So we're starting our first meeting on Thursday, which is tomorrow actually, 4 to 5 p.m. It's on Zoom, so feel free to attend. And just quickly to answer some of your questions. First of all, how to get accepted into these programs. Really, the most important thing you want to know is who is the program trying to serve? Like, are they trying to serve people who are, first of all, like community college students? Like if the program is specifically asking for a community college student, then you definitely should apply. Or if they're asking for first generation students or college students, then you're definitely the person to apply. So there's a lot of information in the program description that you should read and see how it applies to you. And then I think that the most important thing for a lot of programs is how motivated you are, how interested you are. That really will show in your essays. And I'm sure there are some resources in your college where you can get help with trying to get that message across, with trying to learn how to write applications. And we also have that sort of mentorship in our program. Unfortunately, we're kind of low on mentors right now. We've made a lot of assignments already, but in the future years, you can apply. We help with transfer application, we help you with writing or strengthening your transfer applications or any other type of scholarship applications. So reach out to us about that. So yeah, motivation. And then for mentors, how do you find good mentors? It's a great question. And I don't know the answer to it, but I think you will know when you have a person that you think is basically looking out for you. They are helping you become more independent, and also they are encouraging you to challenge yourself and kind of aim high. And once right now at community college, or once you transfer, the best thing is really to talk to your professors. Don't be afraid to talk to them, and you'll realize that some of them are really interested in teaching and mentoring, and those are really good mentors because that's actually their passion, that's their interest. So talk to as many people that you can, and you'll notice which ones you would like to, have as a mentor. I'll pass it on to you, Sean.
That was a great answer. It seems like an awesome program. I think could be really valuable. I do think that there are a number of these programs I've been involved with too, just in the past year, kind of working with community college students who have applied to then be placed in labs. I think that can be a great way to get your foot in the door, kind of as Teni was alluding to, applying to a program instead of a lab specifically, I think can really help. And maybe it's a little more structured too, and you might have some kind of seminars and things like that through the program. Touching a little bit on cold emailing labs, I totally agree. I think that just passion is the way to go. I think it's a numbers game. It certainly isn't easy. But I think that, you can help by making yourself stand out if it's something that, you've really done a little bit of homework. And that can be a lot to do for a lot of labs. But I think starting with the labs that you're most interested in, really showing that you've read their website at a minimum, maybe you've taken a look at a paper, kind of just showing that like it's not just that you just want to work in a lab for the sake of working in a lab, but you really want to work in this lab for whatever reason. As I said, I think it takes time to develop a passion. And so you might not necessarily know that like, I want to work in this specific field. Of course, that's kind of would be tough to know right off the bat, but something where you're kind of like, this seems really interesting and I want this opportunity. I think that really helps and can come across. And you might be surprised, I think would differentiate you actually from a lot of people who just email labs, saying, I just want a research position. So I think that can work. And in terms of the mentor, I think, it's, a lot of it comes down to the personal relationship. I think Teni's absolutely right that, having mentors who are interested in teaching and those kind of professors that you might meet definitely are good places to start, because they do, they're really motivated to work with students specifically. But I think, kind of having someone that you have a good personal rapport with can be a great place to start. And so, if that's kind of like even just you might meet a professor and from a class, and then, kind of build up to asking if you can do research in their lab, I think could be a good place to go. But yeah, I think it just kind of takes some perseverance, but certainly doing the homework beforehand can be really helpful.
Thanks so much, both of you. I know that was a lot of different questions to answer at once. You both did a great job. The next question I want to ask came in, what is a typical day for you? I know, Teni, this must have changed since you're now in medical school, but feel free to describe both the PhD and an MD, and sort of contrasting sort of the day-to-day conduct of research, which we all know can be slow, and the payoff can't come until much later. How do you sort of look at the benefits of that lab experience day to day and keep yourself motivated through different interactions?
Sean, would you like to go ahead?
Sure. So yeah, my typical day has been the same. I don't think it's changed that much coming from grad school into the postdoc. It's kind of like science is my job now. So it's, coming in, planning out experiments, you're not working alone necessarily. You're working in a lab with other people, but your project is kind of your own. And so you're kind of driving that forward. And so it's kind of each day is planning out what are the next steps that I'm going to do? What experiments am I going to do? A lot of it is kind of like maintenance and building up for these big experiments, kind of like Panos was saying, it can be a slow process. You have to get all the reagents, you have to make sure they work, you have to maybe make some proteins you need and stuff. So all of this can be slow for maybe in a month, you're going to do the real experiment that you're excited about. But I think you get by with kind of like the little wins here and there. You're always doing some experiment that's asking a question every day. And I think, sometimes you see something or you discover something that only in that moment. That's kind of, I think, the thrill of doing research is that, sometimes it might not be a big question, but you are the only one who knows that when you make this change, this happens to those cells. And then, you kind of go through and build on that. I think that's really the fun part. So I think it's a mix of that. Of course, there's also meetings and sometimes there's some teaching as well. And then if you're working with kind of a mentee, then you might also have someone who's kind of working under you that you're also training. So I think it's definitely a mixed bag. Each day is different, certainly, but it is a lot of fun.
Yeah, so the grad school basically is very self-driven. You don't really have a set schedule. You're kind of told that you have 4 to 5 years to make a project work. It took me 7 years, and I'm still working on it. And you never know, basically. But that shouldn't be unless like you really, really want to get out of school right away, because you only go to grad school if you're actually really passionate about it, about science, not necessarily school itself, but about the science, about finding out answers to questions and finding out how things work. So just throwing this out there that really a lot of people who go through this, they try to enjoy the journey. And some days it's really hard. There is a lot of failure involved in grad school. A lot of the times your experiments don't work and you just have to go to bed at night and get up the next day and start over. And it's really important to build that resilience, to know that it's okay. I failed today. That's totally okay. I'll start over tomorrow. Maybe it'll be better. Might not be better, but at some point it will be, you will find out something. And like Sean said, you're going to be the only one in the world that knows the answer to that question. And that's just like the best feeling in the world. So related to that, there are also other types of failures that we face in our lives. Like if a class didn't work out so well for you, those things shouldn't keep you from pursuing something. Like I said, I was really bad in biology in high school. I ended up doing a got a PhD in biology, and now I'm pursuing an MD. So like it's all now all I work with is biology, but sometimes classes are not reflective, or grades are not reflective of how much you enjoy a certain topic. I'm going really off topic here, but that said, the medical school curriculum is very different from grad school curriculum. There's a lot of structure to it. We have classes pretty much all day, and there are deadlines that you have to meet. So there's a lot of objectives that you have to meet at certain times and days in your education, and it's all laid out for you. So the structures are very different, and I'm happy to talk more about this some other time or individually with anyone who's interested about differences in grad school and med school.
Thank you so much, Teni and Shah. I know we're coming up to the end of the hour. We did have one quick question that came in when you talk about reaching out to labs. Does that include labs at institutions you are not a student of? Is that a barrier? Or can students from one college university also join a lab somewhere else? I think Teni touched on the program that you're running briefly, and Olivia put the link in the chat. And I think that could help with this question. Teni, is there anything you'd like to add on this point about working at other institutions?
I think it's totally fine wherever you want to apply to. I don't know. What do you guys think, Panos and Sean?
Yeah, I think it can be open. Oftentimes it can just be kind of what the, there might be some regulatory element with the university, but I think oftentimes it's kind of the PI and what they want to do. And I think that certainly if you're passionate about it, there's nothing that I think could prevent you from working in a lab at a different institution. Absolutely.
Great. Well, thank you again to Sean and Teni. This was wonderful. And again, make sure to pick up the link that Olivia put in the chat. And I'll hand it back to her in case there are any closing words she'd like to share.
Yeah, thank you so much, Sean and Teni. Thank you, Teni, for sharing that information. I hope that, you guys go and click on the link and join this new program that we're starting. I also wanted to remind you that we have 2 more events coming up in October 20th, and the other one I believe is in November 3rd. So be on the lookout for information to join us the next time. Thank you.
Thank you, everyone.
Bye.